So some big differences between cardiac and skeletal muscle fibers. Some big differences between cardiac muscle and skeletal muscle fibers. So cardiac muscle fibers, they have what? One, maybe two nuclei per cell. Skeletal muscle fibers, multinucleate. Cardiac muscle fibers may branch. Skeletal muscle fibers, they typically don't. Cardiac muscle fibers have loose T-tubule sarcoplasmic reticulum connections. We already mentioned that. Skeletal muscle fibers have tight ones. They butt right up against one another. Cardiac muscle fibers have less sarcoplasmic reticulum, which means less calcium in the cells. Skeletal muscle fibers, by comparison, have lots of sarcoplasmic reticulum, so plenty of calcium. Here's an important thing to jot down related to that. I'm even going to change colors, so maybe it'll stand out a little bit in your notes. Let's go with blue. No, I'm already using blue. Let's go with pink. That would work. In cardiac muscle contraction, somewhere around 20% of the calcium needed comes in from the ECF, extracellular fluid. Extracellular fluid, or from outside the cells. Now I'm going to pause here and I want you to think. Way back when in 223, and you talked about the function of, say, your average multipolar neuron. Oh, this is a bad looking neuron, but I think you get the idea. So there's neuron. Wasn't calcium needed here, but almost all that calcium came in from where? Outside the cells, like that. In skeletal muscle physiology, we needed calcium, but the calcium all came from one point inside the cell to another, from the sarcoplasmic reticulum into the cytoplasm or sarcoplasm of the cell. In cardiac muscle fibers, most of the calcium, 80%, comes from the sarcoplasmic reticulum out into the cell, but a significant amount, 20%, comes in from the outside. So I want you to look at this and this. Do you see why people in the future, and I mean the people who hear my voice right now, do you see why people might freak out, be hyper alert, hyper aware of calcium ion levels in the extracellular fluid when we're worried about nervous function or heart function? Because for these two tissues, calcium enters the cells from the outside, some that we have. So that's a big difference. These loose T-tubule SR connections, which means slower contractions, and less sarcoplasmic reticulum, less calcium, I have to rely on a significant amount of calcium to come in from outside the cells. This is sort of a big thing. Also, cardiac muscle cells, compared to skeletal muscle cells, they have really, really big mitochondria. Cardiac muscle cells have huge mitochondria. I'm going to continue below here so you can see. Cardiac muscle cells have very large mitochondria in the cells. They're great at aerobic metabolism, terrible at anaerobic metabolism. Cardiac muscle cells I'm talking about. This is why when we have a lack of oxygen, aerobic metabolism, when we have a lack of oxygen, we get very worried about neurons and cardiac muscle cells because they really can't do anaerobic at all. Worth a darn. Great big giant mitochondria to do beautiful aerobic metabolism, but very, very poor at doing anaerobic compared to other tissues. We also have, I'll put it this way in your notes just so you're scientifically accurate. Little or no regeneration of cardiac muscle fibers. Little or no regeneration. So remember, this is where we have the cardiac. We're continuing from above, right? The cardiac muscle fibers. Little or no regeneration. I say little. There might be a tiny bit of regeneration of cardiac muscle that physiologically or cytologically speaking is from migration of cells from one point to another. Tiny, teeny weeny, teensy tiny bit. I'm pinching my fingers together right now. I know you can't see it. Pinching my fingers together, teeny weeny. Here, let's go. I'll be all techie here. Let's go with the tech. A teeny tiny bit of regeneration, not enough to be significant. So generally speaking, we think of cardiac muscle cells the same way we think of neurons. If you lose one, it's gone. You're not going to replace it. So much like nervous tissue, we're talking about extremely limited regeneration in the extreme. So really not any regeneration of these tissues. But you may have noticed I have not hit upon the biggest difference, perhaps the largest difference that people think about when we talk about the differences between these two muscle fibers types. The biggie that I didn't even say anything about is what? The intercalated discs. The intercalated discs. That's the big one everybody always focuses on. But somebody, once upon a time, probably told you the following. You really need to know what an intercalated disc is. If you don't, you're going to have trouble in the first part of Bio 224. Now, how often can somebody say the following? The day has arrived. The future is now. This is the moment where you really need to understand what an intercalated disc is, or you're going to have some trouble. Intercalated discs are collections of gap junctions and desmosomes that connect neighboring cardiac muscle cells. I'm going to say it again. Collections of gap junctions and desmosomes that connect neighboring cardiac muscle cells. This is a big thing. This is a huge thing. If you don't remember what a gap junction is, I suggest you dig out those old 223 notes, take a look at them, And what's the phrase? School yourself a little bit because you have to understand what's going on here. A gap junction. So here it is, and I want you to put this in your notes. I'm suggesting you draw a picture, which means you get out the colored pencils, you get out the highlighters, the super fancy clicky pen with the different colors in it. I don't care what you're using, but get something with some different colors. And I want you to make this picture. I want you to create this picture that I'm going to make right here. Yes, I know you can just look at this video and stream it and watch me. That's great, but this is something you're going to want to have. So make this picture with me. And let me draw for a little while, and then you can pause and catch up. So I'll use two different colors. What we've got is two neighboring cardiac muscle cells. Cardiac muscle cell A, cardiac muscle cell B, sitting right next to each other. And where they're connected is going to be right here. By what? Well, a bunch of desmosomes, spot or button desmosomes. I think you remember those, like protein stitches, connecting cells, allowing for a lot of stretch. Great. Also, we have the gap junctions. A gap junction is when two proteins in neighboring cells fuse together into one globular protein that forms a channel, a tunnel between them. I don't know how well I'm going to be able to draw this here. The whiteboard tends to work a little better. In fact, probably what I should do is this. Let's see if I can fix this up a little bit for us. Let me take away my two cell membranes. I will redraw them, hopefully, knock on wood, a little better. So here's the cell membrane of cell A. And here's the cell membrane of cell B. Maybe this will make it a little bit more clear. And what happens is proteins found in the cell membranes of these two respective cells fuse together but form a tunnel, a canal. Leave an opening between these two neighboring cells. How's that? That's pretty nice for me. I have to qualify that. For my artwork, that's not too bad. So this gap junction this is fused cell membrane proteins that form an ion channel. That's a key thing to remember from 223. They form an ion channel, a tunnel from cell A to cell B. Now, let me ask you a rather silly question. Somewhat silly. Maybe not too silly, but a little silly. Okay? Here comes my question. I'm looking you right in the eye through the webcam. Does the movement of ions within a cell through a membrane have anything at all to do with action potentials? Think about that. Does the movement of ions through cell membranes, relate it all to the transmission of action potentials. I'm pounding on my own desk here. Of course it does. That's the whole darn thing, isn't it? That's the whole giant stinking thing, is the movement of ions from one cell to another. And what I'm telling you right now is that the way cardiac muscle cells are connected to each other is by ion channels. Hopefully right now you're having the first of a whole bunch of what I call aha moments in this lecture. We can't always have those. Aha, you should be saying to yourself. And you're sitting in the privacy of your own home, watching me or listening to me in this video. So I just want you to let it rip. Let the nerd flag fly, people. And when you feel the need to say, aha, you go ahead and pause this video and say to yourself, aha. Perfectly okay to do that. In fact, I want you to do that. So here's what I want you to think about and have those aha moments. Let's say that cell A is experiencing an action potential. What would be happening to cell A if it's in the throes of an action potential? Well, a whole bunch of sodium would be moving around, right? Wouldn't sodium be flying into this cell through open ligand and voltage-gated sodium ion channels? Right? Wouldn't calcium be getting let loose? How about orange? Wouldn't calcium be getting let loose from the sarcoplasmic reticulum out into the cytoplasm? And for cardiac muscle cells, wouldn't some be coming in through the outside through actually what are voltage-gated calcium ion channels? Right? My point being this. Inside the cytoplasm of cell A, inside the cell A, wouldn't I have a bunch of sodiums and calciums floating around loose in this cytoplasm? If cell A is experiencing an action potential, I'm going to have all these positive ions, sodium and calcium, in here flying around all over the place. And yes, say, aha, you can see where this is going. This is the tricky part of the physiology. The fact that cell A is connected to cell B, and you can tell by the sound of my voice, I'm all excited by this. I'm a ginormous A&P nerd. I am the first to admit it. And guess what, people? So are you. You just hide it better than I do. I'm not even trying to hide it. Don't even attempt to hide it. Love this. If cell A is experiencing an action potential and sodium and calcium are flying around all over the place inside cell A, and cell A is connected to cell B by a bunch of ion channels, aha, see, aha, if they're connected to each other by a bunch of ion channels, what will these ions do? If you go way back to 189, and I want you to give a tip of the old academic hat to your 189 professor, don't you understand diffusion? Things will move from an area of high concentration to an area of low concentration unless you have something in the way to stop them. These doors are open. These are non-gated ion channels right here. Nothing stopping these ions from moving from an area here in A where there's a lot of them to an area here in B where they're not all loose. So calcium, sodium, they will travel through these open ion channels entering cell B. Sodium and calcium ripping around inside cell A will now enter cell B, and this influx of positively charged ions all showing up right in here creates what? A local or a graded potential in cell B. And if enough sodium and calciums come flying into cell B to reach threshold, what does cell B do? Has an action potential. This is a big, big deal here. The fact that cell A is experiencing an action potential can then, let me emphasize because it's my picture and I can make a mess out of it all I want, people. Do you understand how cell A having an action potential can make cell B have an action potential? And then cell C have an action potential. And cell M have an action potential. Because I don't want you to think of this in 2 dimensions, which is the only way I can really draw it. Wouldn't this be, everybody, rotate 90 degrees, all these cells in cross-section all connected to each other by gap junctions everywhere. So if cell A experiences an action potential, that could cause cell B to experience an action potential, C all the way to Z experience an action potential just because cell A did. Just because cell A did. These intercalated discs, these gap junctions, allow for the transmission of action potentials from cell to cell to cell in cardiac muscle. This is not the same as skeletal muscle. There are a couple things that we in the science nerd biz like to say here. Jot this down in your notes. It's a good quote to have. Electrically speaking, cardiac muscle behaves as a single unit. Cardiac muscle behaves as a single unit. Meaning, if one of them contracts, then they all contract. So the behavior of the cells is very much like skeletal muscle fibers. When A has an action potential, sodium rushes in, get a local or graded potential that reaches threshold, the whole thing contracts, sarcomere shortens, la 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 la. But I can communicate that action potential to cell B and cell Z and all the others because the cells are connected to one another through gap junctions. So this single unit thing. The action potential would be slower because of the loose T-tubule SR connection, but the action potential can travel from cardiac muscle cell to cardiac muscle cell through those gap junctions. This is the way we can communicate the signal to contract. And think of this, I have to move away from it so I can ask you a question. This might help reinforce the idea that I hope you're getting here. So, if the action potential can be transmitted, if the action potential can be transmitted, just hit a button on my pen, I got to fix that, sorry. If the action potential can be transmitted from one cardiac muscle cell to the next, from one cell to the next cell through the gap junctions, then how many cardiac muscle cells must be innervated or controlled by a neuron? If the action potential can travel from one cell to the neighbor, then how many cardiac muscle cells must be controlled by a motor neuron? Think about it. I'll give you a couple minutes. I don't mind an uncomfortable silence. How many would you have to control or spit on with a neurotransmitter to make them all contract if they behave all for one and one for all like the Musketeers? The logical answer that you probably come up with, Mr. Spock, is 1. The real answer is exactly 0. What, you may say? Question mark, exclamation point, exclamation point. It would be logical that I only need to control one of these cells, let's say A, by spitting a neurotransmitter on it. Stick with me. I'll just make a mess out of my picture. Again, I can do that. So if a neuron came down here to spit on cell A to initiate an action potential, then cell A has an action potential, cell B, cell C, cell Z would have an action potential. Perfectly logical. That's the logical answer. I would need one neuron. The real answer is I don't even need that neuron. Don't need it. Real answer is 0. That's weird. We'll get to that in just a minute. The real answer is 0 of these cells would have to be controlled by a neuron. Now, we have a slower contraction, maybe about half a second for a cardiac muscle cell, which is an eternity compared to skeletal muscles, but it's still an action potential. How do I get this 0 cells need to be controlled by a neuron? Well, that brings us to the idea of autorhythmic, we call them, autorhythmic cardiac muscle cells. Autorhythmic cardiac muscle cells. They have their own rhythm. Autorhythmic cardiac muscle cells. So let's put this in our notes. About 1% of your myocardium, about 1% of the myocardium are these autorhythmic cardiac muscle cells. They will contract on their own without innervation. These cells will contract on their own. No neuron required. There's some terms that physiologists use to describe this. I'm going to write them down because you hear them all the time. They'll say that these cells spontaneously depolarize. They'll say that these cells spontaneously depolarize. That's a common term you hear. Or they have an unstable cell membrane. Or they might even say both. I'm not sure I like either one of these terms. Let me tell you why. Let's say we have this cardiac muscle cell right here. Like that. We have to think about the cell membrane of this cell. Cell membranes aren't just straight lines like that. We know better. The cell membrane has proteins embedded in it every so often, functional proteins, right? We remember that. Channels, tunnels, receptors, all that sort of stuff you've studied in coursework before you ever heard of me. These particular cells, these autorhythmic cardiac muscle cells, their cell membrane, like all cell membranes, has holes in it, channels, tunnels, like that. Let me try to draw this in so it makes some sense for you. Do you remember from back in 223 a discussion of non-gated ion channels like we would find in a gap junction that we just talked about? Well, these autorhythmic cells have non-gated sodium channels as part of their cell membrane, more than other cells have. They have non-gated sodium ion channels. So that means sodium that's out here tends to leak into these cells at a higher rate than is normal. Let's say these green things are non-gated sodium ion channels. So sodium enters these cells faster than the old sodium-potassium pump can kick it out. So I keep getting this gradual buildup of sodium in here, causing the cell to experience what? An action potential. So these cells, here I'm picking on the terminology you hear so often, they don't spontaneously depolarize. There's nothing spontaneous about this. They don't have an unstable cell membrane. They just have a different cell membrane than other cardiac muscle cells. So if you give them enough time, sodium will come in here sufficient to reach threshold, causing the cell to experience an action potential. No neuron required. If you take these cardiac muscle cells and just let them sit there long enough, what will happen? They'll have an action potential. If they just sit still long enough, they will experiencing an action potential. That's what these cells are all about, these autorhythmic cardiac muscle cells. Again, feel free to pause, rewind me, play this over again. It's not so much that their cell membrane is unstable and they don't spontaneously depolarize. They just will on their own without a neuron having to open up any doors, because they have open doors as part of their cell membrane, non-gated sodium ion channels more than other cells do. So what makes these cells depolarize? Just enough time that allows sodium to flood in, and they have an action potential, and away they go. If you're back to looking at me, why not play with the technology here? If your heart does not need to be controlled by a neuron, it'll beat on its own. What would happen if I took a pair of surgical scissors and I went into your chest and I cut the nerves that control your heart? If your heart's beating at, say, a steady 70 beats per minute, like that, and then I cut the nerves that control it, guess what happens? Does your heart stop? No. Do you know what your heart does? It speeds up. If you're at a normal resting heart rate, let's say of 70 beats per minute, and I cut the nerves, your heart speeds up.